Assalamualaikum warahmatullahi wabarakatuh. Hi everyone. I will be present the material from one of the journal article that is Biology and Biotechnology of Follicle Development. Before starting, allow me to introduce myself to you all. My name is Khalwa Alfiana. In this presentation, I divide it into 3 points that are introduction, explanation, and conclusion. Point 1. Introduction. Important information on the abilities square oocytes. The study of in vitro development of oocytes give important information on the abilities which square oocytes during their early follicular stage in vivo. The next variation of follicle number. Recently, it was demonstrated that the inherently high variation in follicle numbers during follicular life is also associated with significant alteration in infrafollicular estradiol production, which is the hallmark for follicular function. For follicular function, an expression of K genes important for differentiation, function, and survival of the granulosal and cumulus cell in the large tree follicle growing during follicular wave. Process of germ cell. That is the picture how the process of germ cell formation. The steps are 1. Generation of primordial germ cell in the yolk state. 2. Migration of primordial germ cell to the genital rack. 3. Colonization of the gonad by the primordial germ cell. 4. Differentiation of the primordial germ cell to oogonia. 5. Oogonial proliferation. 6. Meiotic initiation. 7. Arrest in the diplotent stage of meiotic prophase. Primordial and primary development of the follicle. At present, the oocytes is considered to play an important role in follicular organization during the process leading to ovulation. It is assumed that the oocytes control the proliferation of granulosa cells and later their differentiation into asteroid and protein screening cells. On the other hand, granulosa cells are indispensable for oocyte growth, differentiation, meiosis, cytoplasmic maturation, and control of transcriptional activity within the oocytes. When the oocytes reach a certain size threshold, it secretes factors that inhibit the ability of granulosa cells to promote oocytes growth. This indicates the oocytes determine not only the growth of the follicle but indirectly also its own growth. And then cell interaction at the oocytes granulosa cell interphase. The cell and cell extracellular matrix chemical interaction affect the hormone production and the expression of growth factor of each cell compartment of the follicle. The interaction also consolidates the differential function of germinal and somatic lines of the follicle and also coordinate the process of oogenesis and folliculogenesis. Since the oocyte development is a critical aspect in the follicular development, and the oocytes exert profound effect on the granulosa cell. The interface between oocytes and granulosa cell emerged as the most significant control sites in the coordination of follicle development. Autocrine and paracrine regulatory factor. The autocrine and paracrine factor involved in follicle growth and differentiation include protein and hormone. The ones that regulate the formation of the primordial follicle have not been completely elucidated, although it is known that neurostrophic are involved as shown by the presets of part of the 5 and ST knot. In this table, we can see oocytes lost during meiosis. Diameter of early follicle and oocytes. The diameter of the primordial follicle varies between 9 micrometers and 55 micrometers and is significantly different among species such as hamster, mouse, pig, and human. The oocyte diameter of the same species of the same follicular stage show significant difference. Follicular diameters converge during primary 14 until 62 micrometers and prenatal stage and then show great disparities during early antral and preovulatory stage when most follicles are compared to hamster and pig follicle. And then kinetic of oocytes development. The oocyte is an extraordinary and fascinating cell because it can program its nucleus and start normal embryonic development, sometimes even without participation of a spermatozoa. Up to now, only a limited number of transcription factors are known to be involved in this process. However, recent progress in this field is significant. 90% of the scientific studies focus on the identification and characterization of transcription factors. Development of oocytes inside the follicle in bovine and Burchan species the capacity to undergo meiosis in occur more gradually after the formation of the anther than in most bovine follicles smaller than 2 millimeters in diameter show a lower mean maturation rate and greater susceptibility to fertilization anomalies that large size one. In a similar way, the in vitro development of blastocysts was lower when oocytes were obtained for follicles smaller than 6 millimeters. Meiotic arrest. Morphology classification of MAA generally denotes an oocytes that a mature having arrest in metaphase 2 and is presumed to be capable of fertilization. In most mammals, meiosis and all oocytes initiated during fetal life but stop prenatally in the This Jotan stage of the process, the meiotic arrest is maintained until follicular development to a Raphian follicle. During that time, the oocyte growth adapts itself with that of follicle cell, which interacts through the effect of gonadotropin and steroid hormone, as well as with other intrafollicular molecules such as growth factor. And the last part, conclusion. Follicular growth during the parental early antral transition. Follicular growth during the parental early antral transition is mainly regulated by intraovarian oocyte granulosa, the cell interaction and regulator such as growth factor, cytokinesis, and steroid. The follicle must remain intact in order to preserve normal function. Actually, there is a strong prospect for the production of molecular tools to classify development potential. And then cytokinesis and growth factor also promote follicular survival and growth. Cytokinesis and growth factor also promote follicular survival and growth during the parental to early antral transition by suppressing granulosa cell apoptosis and follicular atresia. Those, for example, GDF9 enhance parental follicle growth by upgrading the cast cell androgen production. Assisted reproduction technologies. Assisted reproduction technologies involve controlled follicular development and exogenous hormone stimulation of oocyte maturation. Nevertheless, a successful oocyte in vitro maturation would eliminate the need for hormonal stimulation used in ART. In addition, by understanding the molecular and cellular mechanism in the control of follicular development during the parental to early antral transition, it is possible that EVM protocols could be developed to provide the signaling machinery necessary for oocyte maturation competency. Alright, this is my presentation. If you have any question, please raise hand or you can contact me later on contact available. Thank you for your kind attention. Please forgive me if I have some mistakes. Balas I say wassalamualaikum warahmatullahi wabarakatuh.